Okay, so first of all, I would like to thank the organizer for organizing this great meeting and putting it together. And it's a great honor to be one of the speakers among so many great scientists today. So yeah, I would like to use this opportunity to tell a little bit about metal and metalloid. Oops, sorry. I'm just trying to make it full screen. About metal and metalloid dependent anaerobic methane oxidation, and I will focus mainly on the iron as electron acceptor and focus on our field study in Vietnam. And this was the work that I've done during my PhD at the University of Tübingen. But I would like to also use this opportunity to briefly talk about other alternative electron acceptors for methane oxidation. So yeah, let's start with the iron as electron acceptor. And so the main motivation for this work was related to the fact that arsenic groundwater contamination is a very serious problem worldwide. Recent study was shown that up to 220 million people are potentially exposed to arsenic groundwater contamination, overpassing the safe limit of 10 micrograms per liter. And what is particularly concerning is that majority of these affected regions are in Asia, where people often do not have access to water treatment facilities. And in consequences, a lot of people are actually consuming this highly contaminated water. And obviously this is very problematic because arsenic is a toxic metalloid. It's a human cancerogen, leading to many health issues, mainly expressed in a skin disease, leading to eventually cancer. And so because of this serious contamination of water in this part of the world, last 2 decades brought really lots of research focusing on trying to understand what is the mechanism of methane, of arsenic release from a sediment to the groundwater. And so there was a really lots of study done to really understand this process. And yeah, we know, for example, that oxidation of arsenic-rich sulfides, such as pyrite, that has a very strong affinity to arsenic, is one of this mechanism. Also, competitive sorption of arsenic with phosphorus is a second. I will just find a pointer option here. And yeah, so arsenic and phosphorus have a similar structure, and therefore they compete for a sorption site on the iron oxides. So that is also one of the mechanisms of arsenic mobilization. Changes in sorption capacity of iron hydroxide, usually due to the changes of pH of water. But really one of the most important process, and the one that I've been focusing a lot on, is microbial reductive dissolution of arsenic-bearing iron-3 minerals. And this is the process that I've been studying quite extensively during my PhD at the University of Tübingen. And well, this process is fundamentally very simple. So the way it works is we have iron-reducing microorganisms that can use pretty much any type of organic carbon, the organic carbon, preferentially short-chain fatty acids, oxidizing it to carbon dioxide and dropping electrons on iron-3 minerals that retain arsenic. And by reducing this iron-3 minerals, they co-mobilize arsenic that is bound to this iron minerals. And so, yeah, we do know actually quite a lot about this process. We know about the microorganisms that can mediate it, among others, Geobacter, Shawanola, Geotrix, many different fermenting microorganisms also. And so we know that the dissolved organic carbon play a very important role in this process. But in fact, in our study site in Vietnam, in a small village of Van Phuc, about 15 kilometers south from Hanoi city, and we realized that when we were monitoring this, monitoring wells that we have, that in fact there was not that much organic carbon available in these waters. Actually, it never really exceeded 7 milligrams per liter, but methane was very, very abundant. And we realized that actually there is some positive correlation between high methane concentration and high iron and arsenic concentration. And this was something that we really observed in most of the wells, where methane was abundant and was highly available. And then also we saw the increase of iron and arsenic. And that wouldn't be really that surprising, because actually we know that strongly methanogenic conditions are associated with a strongly reducing condition. And this can trigger reductive dissolution of iron minerals. So that could be potential explanation why we have also high iron and arsenic. Also, because when we have a methanogenesis, usually this is linked to the quite intense carbon turnover, and we have when we have lots of carbon turnover, carbon degradation, lots of short-chain fatty acids available potentially. This could also stimulate heterotrophic processes, and obviously iron-reducing microorganism would thrive there. So that would also explain why iron and arsenic would be abundant at the sites. So probably I would not really continue into digging into the subject, if not a couple of papers that came out and that I read that show that actually methane-dependent anaerobic methane oxidation can be linked to the reduction of iron. And there are a couple of microorganisms that have been shown that can do it. In this paper specifically, they were talking about ANME 2 archaea, anaerobic methane oxidizing archaea, affiliating with Candidatus methanoperandin species. And so, first thing that I wanted to see is whether these microorganisms can be found in our field site. So I look at the microbial community, both in sediments and in the groundwater. And it was quite striking to see that, in fact, microorganisms involved in a methane cycle were way more abundant than microorganisms involved in an iron cycle. And what was particularly interesting and exciting was that indeed Candidatus methanoperandin, we could pretty much find it in most of the wells we were monitoring. We wanted to test this hypothesis whether anaerobic methane oxidation can be a link to the iron reduction and arsenic mobilization. So our hypothesis was quite simple. Methanogens that are producing methane in our aquifer, whether via hydrogenotrophic methanogenesis or acetoclastic methanogenesis, and can supply substrates for anaerobic methane oxidizing archaea that would use it as an electron donor, and then dropping electrons on the iron and reducing iron similarly to iron-reducing bacteria, leading to a co-release of arsenic to the groundwater. The way to test this hypothesis was to set up incubation experiment. We did a drilling campaign during which we collected sediments from 30-meter depth, and I supplemented these sediments with a methane. And the experiment lasted quite long, over 220 days. And during the first 125 days, I didn't see much happening. Actually, only about 6% of iron was reduced. Mainly, it was observed in sediment. So there was formation of iron 2 directly in a sediment, but there was no iron being released to water. However, there was still some arsenic being mobilized to the groundwater, to the artificial groundwater. And that was probably linked to the fact that this arsenic was rather loosely bound to the iron minerals, and even a small reduction of these iron minerals would lead to the release of quite a substantial amount of arsenic. And so at that point, I thought that not much will happen anymore, and I kind of stopped sampling this experiment. I didn't put much hope into it. But eventually, after a while, after 30 or 40 days, I realized that the bottles started to change the color. The one that were supplied with methane become more grayish brown. And I immediately knew that this is related to the reductive processes taking place. So I restarted sampling these bottles. And indeed, I observed that iron 2 actually quite substantially increased over this period. We observed a quite intense reduction of iron in the sediment, and also release of iron 2 to the solution, which was positively correlated also with the increase of arsenic in the groundwater in the solution. Overall, at the end of the experiment, after 220 days, we saw that up to 25% of iron was reduced, and a significant amount of arsenic was released to the water. So that was quite exciting, because really this already showed that methane is driving this iron reduction as the only electron donor and only carbon source. Clearly, it stimulated reductive processes. So we wanted to know, of course, what microorganisms are involved in this process. First thing we've done is to look at the bacteria and archaea 16S gene copy numbers, as well as an MCRA and PMOA genes. So these are the genes that are commonly used to target anaerobic methane oxidizing archaea, and PMOA for methanotrophic archaea, usually aerobic one. After 125 days, we saw that bacteria community dominated over archaea, and there was much higher abundance of PMOA gene over MCRA gene. This changed quite a lot in the end of the experiment, when we saw that there was this more intense reductive processes happening. And then we saw that archaea were taking over, and also MCRA gene was more abundant over PMOA gene. So that was already quite promising because, yeah, archaea were associated with these reductive processes previously, and so we were hoping to see that. And then in the end, we also confirmed that with the 16S amplicon sequencing, which in the end ultimately confirmed that, yeah, archaea were responsible for this process. So during the first 125 days, we enriched a microorganism called Metabologia aureusea. And we believe that that was the microorganism that was responsible for this initial 6% of iron reduction. But ultimately, in the end of experiment, when there was really intensive iron reduction happening, Candidatus methanoperendens was highly enriched. And that was really exciting because this was the organism that we were really targeting and that we were hoping to see. So yeah, at this time, this Candidatus methanoperendens account for about 60 to nearly 70% of microbial community. And we were pretty positive that this is the microorganism that is actually driving this process. So we decided to look into a metagenome also. We picked up just 2 samples to look a little bit deeper into it. And that was done by our colleague, my current colleague from my current lab, Paula, that analyzed the metagenome. And well, she indeed confirmed that the microorganism that we saw in my amplicon sequencing, such as Metalogia, carry PMOA gene, so the gene involved in aerobic methane oxidation, particularly methane monoxygenase. Although the setup was aerobic, it seems like some kind of aerobic methanotrophs were still thriving in this setup for some reason. And this is not uncommon because we often see this aerobic methanotrophs in anaerobic environments. We still don't know really what was the electron donor for these microorganisms, but yeah. And then we had, of course, the Methanoperendens species. In fact, there was 5 different Methanoperendens strains. All of them, of course, encode for the metal coenzyme A reductase. But what was particularly interesting is also the fact that they all encode for arsenate reductase. And that was quite intriguing. They all seem to be resistant to arsenic and likely also able to use arsenic as electron acceptor. And that was later published in a paper by this Chinese group that indeed confirmed that arsenate specifically can be used potentially as electron acceptor in anaerobic methane oxidation, and that was shown for the wetland soils. But one last piece of story that was missing in this study is to really confirm that methane was actively being oxidized. So we decided to repeat this experiment that was done by my master's student, Alexandra, that set up pretty much similar experiment that I've done previously. We use very similar sediments from similar depth. And we again incubated with methane, but this time we use a label methane. And so, yeah, basically these study again confirmed that iron reduction, so iron 2 formation in sediment was happening in the parallel with the formation of label CO2. And in this experiment, even more iron was reduced. We calculated that about 35% of iron was in total reduced during this 200 and also 20 days about of this experiment. And again, we enriched Candidatus methanoperendens. In the end, and this archaea represented about 60 to 70% of microbial community, which I think pretty strongly indicate that this is the microbe that really is doing this job. So yeah, in the end, I think we really managed to show that anaerobic methane oxidation coupled to iron is quite an important mechanism that leads to the arsenic mobilization in these aquifers. And we believe that this mechanism is not only relevant in this specific site, but in many sites where methane is abundant. And it can certainly lead to the quite substantial release of arsenic to the groundwater. But now there is quite a lot of study coming out, and we know that actually methane-dependent and iron-dependent anaerobic methane oxidation is actually happening in many different freshwater ecosystems also. And therefore, lots of study are happening to look into it a little bit deeper. as it is a potential sink of methane. But we need to keep in mind that methane can also serve as electron donor for other electron acceptors. And so this will substantially affect mobility of arsenic in aquifer. So for example, in the aquifers where sulfates are present, then methane can be coupled to reduction of sulfates. And this is the process that can be driven via the consortia of archaea and sulfate-reducing bacteria. Via this process, sulfides are going to be formed, and this can lead to the precipitation of iron sulfide or arsenopyrites, and that could be a potential sink of arsenic. This could be very relevant for aquifers close to the coast where there is maybe input of marine waters. On the other hand, we have processes such as manganese-dependent anaerobic methane oxidation that we know that can also happen. And in this case, if manganese is used as an electron acceptor, this can lead to the not only mobilization and release of manganese to the water, but because manganese is also a very powerful oxidant, it can also release arsenic that is abiotically oxidized by manganese. And now we also know that arsenate can serve as electron acceptor directly. And so if this metalloid is used as an electron acceptor, then it will also increase mobility and likely increase the concentration of this metal in the groundwater. So as we know now, there are actually much wider range of different electron acceptors that can be used in anaerobic methane oxidation. And this is something that I'm now exploring at my current lab in Radboud University in the Department of Microbiology. Because, anaerobic methane oxidation and methane oxidation in general is a very important source, sink of methane. And therefore, there is a really increasing number of studies that look into these processes. And for a very long time, it was believed that methane oxidation can happen only aerobically with oxygen as electron acceptor. And it was only in the late 70s that sulfate-dependent anaerobic methane oxidation was reported for the first time. This process is energetically really on a thermodynamic limits. And in fact, this process is mediated by anaerobic methane oxidizing archaea and sulfate-reducing bacteria. So this energy further need to be split between two syntrophic partners. Eventually, also thiosulfide and elemental sulfur was proposed and showed that can serve as electron acceptor. Then in 2006, in my current lab, it was for the first time also showed that nitrate-dependent anaerobic methane oxidation can happen, and this is energetically quite favorable reaction. Nitrate also can serve as electron acceptor, also very thermodynamically feasible and favorable reaction. And so these processes have been studied quite extensively in past decades. We know quite a lot about microorganisms. In this case, also anaerobic methane oxidizing archaea appear to play an important role in these processes. In the case of nitrate, it's NCT10 bacteria affiliated with Candidatus methylomirabilis that is mediating this process. And eventually, some 10 years later, and then we currently now have quite a lot of data also showing that manganese oxides and iron hydroxides are also very powerful electron acceptors. And we see that these processes are happening in many different ecosystems that provide quite also good energy for the microorganisms. We know also that ANME2 archaea are involved in these processes. We know also that cytochromes play an important role in the reduction of these insoluble electron acceptors. But then there is a bunch of different other metals and metalloids that from thermodynamical point of view clearly can serve as a very powerful electron acceptors. They can provide quite substantial amount of energy, among them selenite, chromite, bromide, selenium 4. You can notice that it provides more than double what oxygen could provide for microorganisms. Arsenate, which is a little bit less attractive electron acceptor, but also antimony, vanadate, and perchloride. So these electron acceptors has been proposed based on the thermodynamics. There are some preliminary studies already published that indeed show that likely these elements can serve as electron acceptor for anaerobic methane oxidation. However, these processes are still very elusive. We don't know what are the mechanisms, what are the microorganisms involved, and so this is something that I'm quite interested in right now. Just beginning of this year, this month, we submitted a review article in which we actually explored this genetic potential of ANME archaea to use these various metals and metalloids as electron acceptors. And in fact, it does look like these microbes are kind of equipped to use such a wide range of electron acceptors. So when it comes to manganese and iron oxyhydroxides, we know that cytochromes play a very important role in there. We know that ANME2 archaea, in fact, encode for a really high number of cytochromes. So it has been shown that they can encode to up to 45 cytochromes, which is a quite impressive number for non-strict iron reducers. Yeah, they actually, there are some papers also that show that these cytochromes are highly upregulated and expressed when ANME archaea are incubated in the presence of manganese and iron oxyhydroxides. Several genomes of methanoperendin species also encode for a putative selenate reductase that will be involved in a reduction of selenium-6 to selenium-4, but apparently also to elemental selenium. Majority, or if not all, genomes of candidatus methanoperendins encode also for arsenate reductase. And arsenate reductase, of course, is involved in a reduction of arsenic 5 to arsenic 3, but it has been shown also that it can be involved and stimulate a reduction of selenium antimony and also a reduction of perchloride to chloride. And also all available genome of ANME2 archaea encode for a nitrate reductase, which of course mediate the reduction of nitrate to nitrite, but also has been shown to mediate a reduction of vanadate, bromide, and perchloride. And a couple of genomes we also found that there are enzymes involved in a chromate reduction. And so likely some of these methanoperendin species are also equipped to reduce chromite. So in fact, it does look like ANME2 archaea are somehow skilled to use these metals and metalloids as electron acceptors. They seem to be equipped to do so. But whether this is happening via this enzymatic reaction, we still don't know. There is no laboratory study that could really confirm that yet. Another way in which ANME2 archaea can be involved in this process of metal-dependent anaerobic methane oxidation is via direct interspecies electron transfer, about which we probably are going to hear some more today. And this can happen via nanowires, conductive pili, or also outer membrane cytochrome. So in this case scenario, ANME2 archaea would be only involved in the oxidation of methane to carbon dioxide. And electron gains in this process, they will be transferred to metal-reducing partners via these structures. It has been also shown that ANME2 archaea encode for ACD gene. So this is the gene that is catalyzing conversion of acetyl coenzyme A to acetate. So it appears also that ANME2 can, in fact, produce some kind of volatile fatty acids, such as acetate. And if this is really happening, then we can also assume that these fatty acids could be later used by metal-reducing bacteria to further reduce this metal oxides. But again, this is also just a hypothesis, and we do not have yet any evidences to show that these processes are really happening. And so the reason why metal-dependent anaerobic methane oxidation is really interesting also, and I think worth studying is because of its environmental and health implication. Because all of these potential electron acceptor, well, except iron that isn't really toxic, but clearly doesn't improve the quality of water when it's in high concentration. But yeah, all of these electron acceptors are quite toxic, and they have been shown to have some mutagenic, cancerogenic effect on the human health. And therefore, following World Health Organization, and it should not, the concentration of these elements in the water should not exceed certain thresholds. And so if M-DAMO could be indeed achieved, and we could really show that this process is working, that could really provide a very interesting potential bioremediation strategy, because in this process, most of these elements, upon reduction, they would precipitate, that they would form nanoparticles, nanominerals that could be easily removed from the solution, from the wastewater, for example, and therefore it could help in purification processes. Also, in the case of bromide, and the reduced form is less toxic. And so the process of M-DAMO could be also quite attractive, because also methane as an electron donor would be much cheaper than the conventionally used electron donors in the wastewater treatment, such as acetates or alcohols. And moreover, it could be produced in situ via anaerobic digestion. So there is quite a potential for this process in the remediation strategies. But we need to keep in mind that in the case of arsenic, manganese, and iron, if M-DAMO could be linked to these processes, or it is actually what we know already for manganese and iron at least, then it would increase mobility of these elements and likely threaten the quality of water and health of humans. So for arsenic, we know that trivalent arsenic is much more toxic and mobile and more bioavailable for a cell, so that could lead to a potential toxicity. For a manganese oxide, of course, it would release the manganese to the solution, to the water, but also because, as I already mentioned, manganese oxides are quite strong oxidant, and they are often controlling the mobility of different heavy metals in the sediments or soils. And so upon reduction, it would likely also increase the mobility of these heavy metals, in this case, arsenic. And iron oxides, of course, we know that they are having very strong affinity to absorb different heavy metals, such as arsenic, cadmium, chromium, and copper, and so on. And once these iron oxides, oxyhydroxide, would be mobilized, reduced, also these heavy metals would really be released to the water. And that could be also potential threat for quality of water. So this is what I'm currently doing, and we are very lucky in my current lab to have these two enrichment cultures of ANME to archaea, which are very, very difficult to grow, and not many labs in the world are actually able to enrich these microorganisms. We have one freshwater enrichment culture that represent about 30% of Candidatus metanoporandens, and the other one enriched from the rice paddy soils from Italy that contain about 50% of Candidatus metanoporandens. And now currently I'm trying to see whether these cultures are actually indeed capable of using these various electron acceptors, these metals and metalloids, in the process of oxidation of methane, but also to see whether what is the really mechanism, whether it's more enzymatic reaction or via syntrophic relationship with the metal reducing bacteria. And ultimately, I would like to see whether M-DAMO process can be potentially used as a remediation strategy for the wastewater treatment. And so, yeah, as a spoiler, I can say that, yes, I do see actually that these enrichment cultures are very, very skilled in reducing pretty much anything I'm giving them. However, the role of methane and all of this is still not very clear. But this hopefully will be sometimes in the future that I can tell a little bit more about it. So yeah, with that, I would like to thank you all for listening, and all my collaborators in the past and in the present, to Avecta's team that was involved in the Arsenic project, and my current lab at Radboud University. And thank you for your attention.